Good morning, students. I am Shomajit sir. Today I am here to deliver my lecture from the chapter Biotechnology's Principle and Processes. So we'll start a fresh new chapter of biotechnology. Here we'll discuss about or we'll study the definitions of biotechnology and the principles of biotechnology. So watch the entire lecture to get basics out of it. So without any delay, let's start with the chapter Biotechnology, its principle and processes. So let's start. So let's start with the chapter Biotechnology, Principle and Processes. So first we'll see what is biotechnology. So what is biotechnology? Biotechnology is a technique of using live organisms or their enzymes for products and processes useful to human. So what is biotechnology? Biotechnology is a technique which is used, okay, where living organisms are used. Biotechnology is a technique of using live organism or their enzymes for products and process useful to human. So European Federation of Biotechnology, that is EFB, defines biotechnology as the integration of natural science and organism, cells, parts of therefore, and molecular analogues for products and services. Now, what is biotechnology? So, biotechnology deals with number one, microbes mediated processes, that is for making curd, bread, wine. Next is in vitro fertilization, that is test tube baby program. Third is synthesis and using of a gene. Fourth is preparation of DNA vaccine. And next is correcting a defective gene. Okay, so biotechnology deals with what? Biotechnology deals with microbes mediated processes for making bread, curd and wine. In vitro fertilization, that is test tube baby program. Synthesis and using of gene, preparation of DNA vaccines and correcting defective genes. Now, on the principles of biotechnology, we'll see the core techniques of modern biotechnology. First is genetic engineering and second is bioprocess engineering. Now, what is genetic engineering? So, genetic engineering it's a technique in which genetic material, so we know the genetic material is DNA and RNA. So, genetic material is chemically altered and introduced into host organism to change the phenotype. Okay, so what is genetic engineering? Is a technique in which the genetic material, that is DNA or RNA, is chemically altered and introduced into host organism to change the phenotype. Next is bioprocess engineering. So maintenance of sterile ambience in chemical engineering processes for growing desired microbes, that is eukaryotic cell in large quantities for manufacture of antibiotics, vaccines and enzymes. Okay, so in bioprocess engineering it is used for manufacture of antibiotics, enzymes and vaccines. So let's see the basic steps in genetically modifying an organism. Okay, or basic steps in genetically modified organism. First is identification of DNA with desired genes or desirable genes. Okay, identification of DNA with desirable genes. Second is introduction of identified DNA into the host, and third is the maintenance of introduced gene in the host and transfer of DNA to its progeny. So first we'll identify it in gene. Okay, first we'll identify a DNA with having a gene. Then we'll introduce that DNA to the host, and then we'll maintain that introduced DNA in the host and transfer the DNA to its progeny. So first is identification of DNA with desirable gene. So traditional hybridization, that is technique, leads to inclusion and multiplication of undesirable gene and desirable gene. Okay, so it includes, what is traditional hybridization? A techniques used for multiplication of desirable and undesirable gene. Okay, so next is in genetic engineering only desirable genes are introduced. So in genetic engineering only desirable genes are introduced. Next is introduction of the identified DNA into the host. So you can see a bacterial cell, it is having a nucleoid and plasmids. So you can see the recombinant plasmids, then E. coli host cell, and then the transformed cell. So vector DNA such as plasmids is used to deliver an alien piece of DNA into the host organism. So plasmids, that is autoreplicative, that is present in bacteria, that is taken out, and inside that plasmid a recombinant plasmid is made, and this recombinant plasmid is transferred to the host organism. Next is maintenance of introduced DNA in the host and transfer of DNA to its progeny. So you can see a piece of alien DNA has no sequence called, okay, so you can see a piece of alien DNA has number of sequence called origin of replication, ORI, needed for starting replication, so it cannot multiply itself in the progeny cells of an organism. Next, hence the alien DNA is integrated into the recipient genome, so it has ORI, so it multiplies and inherits along with the host DNA. Okay. So first one is a piece of alien DNA. In the piece of alien DNA, there is no sequence of called origin of replication. Okay. So then it's needed for starting replication, so it cannot multiply itself in the progeny cells of an organism. But when the alien DNA is integrated into a recipient genome, so it has ORI, that is origin of replication, then it multiplies and inherits along with host DNA. So next, on the principles of biotechnology, first one is the process of joining and inserting a foreign piece of DNA into a host organism to produce new genetic combinations is called recombinant DNA technology. Next is first recombination DNA, that is rDNA, was produced by Stanley and Boyer, okay, Cohen and Boyer in 1972. So they isolated an antibiotic resistant gene, that is a piece of DNA, from a plasmid of Salmonella typhimurium. Okay, so it was linked with a plasmid vector and transferred into E. coli. As a result, the gene was expressed and multiplied in E. coli. So you can see Salmonella typhi. So by this way, they have inserted the gene. Thank you, Shwan. This was all about the entire video lecture. Here we have started the chapter Biotechnology Principles and Processes, where we have discussed about the definitions of biotechnology and principles of biotechnology. So thank you, and again we'll meet in the next class. Thank you, students.